Mathieu from Houston. He's going to talk about lysosomal admission strategies for the treatment of oxysterol-induced cytotoxicity.
So when I talk about my research, I usually find that most people have a fairly good grasp of cholesterol. They understand that in the cell, cholesterol serves to, it has a condensing effect on the membrane. It also helps decrease membrane permeability. And cholesterol is also involved in lipid raft formation in the cell. However, I find that most people aren't very familiar, at least, with oxysterols. But all oxysterols are oxidized derivatives of sterols. In the case of a human being, that sterol is typically cholesterol, and the oxidation is typically the addition of a single oxygen or hydroxyl molecule onto the cholesterol molecule itself. But the addition of the single oxygen atom onto cholesterol dramatically changes its properties. And it makes it also a much more highly active molecule relative to cholesterol in the cell. The location of the oxidation has a lot to do with the properties that the oxidation confers on the molecule. Oxysterols can be generated enzymatically, and this oxidation normally occurs on the side chain. These types of oxidations normally serve the purpose of increasing sterol efflux. However, oxysterols can also be generated autooxidatively, and these types of oxidations are typically detrimental to the cell, and they normally occur on the steroid nucleus, preferentially near the 7-carbon. And those are the types of oxysterols I've been primarily concerned with, these cytotoxic oxysterols that are modified at the 7-carbon. And these oxysterols are the ones that are associated with many different age-related degenerative diseases, such as atherosclerosis, Alzheimer's disease, age-related macular degeneration, just to name a few. Here's a summary of the effects. Like I said, I'm mainly concerned with the cytotoxicity and inflammatory properties of oxysterols. However, there's a whole list of other things that are involved in, from bile acid synthesis to even cell differentiation. So how do oxysterols, especially 7-keto-cholesterol, how do they cause these effects in the cell? Well, if this is typical cholesterol in a phospholipid bilayer with these phospholipids to the side, you'll see that cholesterol sits rather horizontal between the phospholipids and is actually shielded by the phospholipid heads. However, 7-keto-cholesterol, which would be this molecule down here, has the additional oxygen atom, or the keto group, right here. This keto group wants to interface with these polar regions up above. And so what happens is 7-keto-cholesterol actually inserts at an angle in the membrane relative to what cholesterol would insert at. This increases membrane permeability by pushing apart these phospholipids. It also can result in cholesterol depletion in the cell membrane. So 7-KC increases permeability, it prevents cholesterol efflux, but also limits cholesterol in the cell. And 7-KC can intercalate into normally cholesterol-poor membranes, such as the ER. When it incorporates in the ER, it can cause initiation of the unfolded protein response. So oxysterols accumulate in certain cell types, actually. And what you can see here in the red, this is an antibody to 7-KC. And so you see it's pretty specific, cells that it accumulates in. The red over here is also 7-KC. And we think we know the reason it accumulates in certain cell types. One of the potential reasons is expression of certain receptors, such as in a macrophage, normal LDL uptake for, say, cholesterol, that's regulated by negative feedback mechanism. However, scavenger receptor doesn't contain a feedback mechanism like that, so the cell will continue to uptake oxidized sterol as long as it's available, or oxidized LDL. Typical concentrations of oxysterols in a cell, you'll see about, in a normal cell, maybe 0.1% total sterol can be oxysterol. 7-keto-cholesterol is normally the most predominant oxysterol in a cell, and it's also one of the most cytotoxic. Some cells you can see upwards of 1%. These are in healthy cells. But in a pathological situation, you can have up to 25% of total sterol actually converted to oxysterols. This is rather rare, but this does happen in, say, an atherosclerotic plaque. You'll see 25 to 100 micromolar oxysterols, and this is definitely enough to kill a cell. And they also accumulate in regions where you have maybe lower turnover, such as in the brain, central nervous system. A typical turnover through the body would be about 0.7% total sterol per day, whereas when you go into the central nervous system, it's much slower. You only have about 0.03% sterol turnover per day. And once inside the cell, oxysterols seem to preferentially partition to the endocytic lysosomal compartment. About 45% when they measured 7kc partitioning was located there. Another 40% was located in the cytoplasm. So that's important for our research because we're really concerned about the effects that oxysterols are having within the lysosome. This is a schematic just showing LDL uptake in a cell. And after endocytosis, the endosome fuses with the lysosome. And at this point, it's exposed to certain hydrolases that can cleave the cholesterol esters, or it can cleave the oxysterol esters if they're present. So this releases free cholesterol and oxysterols into the lysosome. And at this point, we can also have a lot of oxysterol formation. And so if we get high enough oxysterol concentrations inside a lysosome, this results in lysosomal membrane permeabilization. And this is just a little diagram kind of showing what's going on. We believe what's happening is, first of all, lysosomes have high iron concentrations. And we believe this is due to uptake of iron-containing compounds. They release the iron in the lysosomes. And then once inside the lysosome, they can undergo Fenton reaction, reacting with peroxide to produce free radicals. These free radicals react with the lipids in the lysosome, causing lipid peroxidation. Enough lipid peroxidation, including 7kc accumulation, results in lysosomal membrane permeabilization. You then have release of the lysosomal contents, specifically the cathepsins, that cause a signaling cascade to occur that results in cell death, necrosis, apoptosis, and necroptosis. So being such a toxic molecule, surely the human body has some way of dealing with it. In fact, we do. There's several enzymes that are known to be active against 7-ketocholesterol. This first one, actually, 11-beta-hydroxysterol dehydrogenase, can interact with 7-ketocholesterol to form 7-beta-hydroxycholesterol. This oxysterol, unfortunately, is more toxic than 7kc. Certain animals, like a hamster, can convert this molecule to the other isomer, the 7-alpha isomer, that is non-toxic relative to 7kc. However, in humans, this has become a target for the treatment of atherosclerosis because it does have an involvement since 7-beta-hydroxycholesterol is more toxic. However, other enzyme reactions result in reduced toxicity. Actually, CYP27A1, which encodes sterol-27 hydroxylase, hydroxylates the side chain, and this almost completely eliminates the toxicity of 7kc. Sulfation also increases 7kc efflux and reduces toxicity, as well as esterification, which results in storage in the form of these lipid droplets inside the cytoplasm. However, there's a problem with all of these reactions. This is a mitochondrial enzyme. Salt is located in the cytoplasm, ACAT as well, ER cytoplasm. And this is an ER enzyme. So all these enzymes are located in regions of the cell that aren't ideally localized to prevent lysosomal membrane permeabilization. Since remember, when we uptake sterols, we uptake them in the endocytic lysosomal compartment, and this is also the point of formation for most or for autooxidatively modified oxysterols. So we studied 7kc cytotoxicity in fibroblasts, and this is a result of a FACS analysis we did. We stained with the Enzymes5 FITC and PI, propydium iodide, and this is an indication of the apoptotic state or not. And what we have here is untreated. So this is what a normal population of cells looks like. These were treated with 50 micromolar and 100 micromolar cholesterol. As you can see, there's really no change in population distribution of these cells. However, when we treat these cells with 50 and 100 micromolar 7-ketocholesterol, we see there's a large shift in population up here, which is a very high increase in the PI signal. And what this indicated to us is that these cells were undergoing a fairly rapid necrosis and probably not apoptosis. So we think it's either necroptosis or necrosis that these cells are undergoing. We also did XTT cell viability assays. And when we introduced 7kc in the cultures, we did it two different ways. Ideally, we'd like to introduce using LDL, but it's kind of expensive, so most people use these sterols just solubilized in ethanol. However, there's a problem with that because as soon as you amend the cultures with 7-KC, it precipitates out a solution, sticks to the side of the culture, so you're not really sure what the concentration that you're getting to the cells is. So in order to try to remedy that, we also solubilize 7-KC in cyclodextrin. So our hypothesis was that the more soluble, more of the 7-KC would make it to the cells, and we'd see a more highly toxic effect. But in fact, we saw the opposite. So this is 7-KC solubilized in ethanol, and we actually have a protective effect with 7-KC that have been solubilized in cyclodextrin. As a matter of fact, it took about twice as much 7-KC solubilized in cyclodextrin to achieve the same effect. These are some metabolites of 7-KC, or just some more oxidized. We had a bile acid and 7-keto-DHEA, which is basically a 7-KC molecule without the side chain. And as you see here, oxidation that we found of 7-KC pretty much eliminates its toxicity. So we had a few strategies that we wanted to look at in order to reduce 7-KC toxicity in cells. And the whole purpose of this is to try to reduce 7-keto-cholesterol concentrations or levels in cells to see if we can help prevent or attenuate some of these age-related diseases, to see the impact that this type of damage has on these cell processes. So our strategy, we thought, would be either to increase 7-KC efflux from the lysosome, or we could attempt intralysosomal transformation of 7-KC to transform it into a less toxic metabolite. And this is the one I'll be talking about mainly today. However, there's a problem with intralysosomal transformations. Lysosomal environments is much different than the rest of the cell. It has a very low pH. There's a lot of proteases in there that would degrade any enzymes we might use. There's also a lack of cofactors in the lysosome, so it kind of ties our hands in what enzymes we could potentially use. In the lysosome, there's one known reductase, so there aren't a lot of redox enzymes, and most of the enzymes that are involved in steroid degradation, either microbial or mammalian, are redox enzymes. So we had to look for enzymes that were able to transform 7-KC, but weren't redox enzymes. Some of the work one of our collaborators did recently was they used they found a cholesterol oxidase that was actually active against 7-KC, and they were able to test this against, I think these were HEK cells. Anyway, what they found was that the diketone formed by the cholesterol oxidase was less toxic than 7-KC, although there was still some toxicity relative to cholesterol. So we thought this was a good starting point to look at. What we actually found was that there was a particular cholesterol oxidase from this one bacteria, Chromobacterium, that lacks this activity, this isomerization activity. And normally well, first of all, I should say normally a cholesterol oxidase is an enzyme used by bacteria to lyse cells, so actually to break open the cells. But what previous research had established is that a normal cholesterol oxidase has an innate isomerization activity that flips this 5-6 double bond to a 4-5 double bond. So this would be a normal cholesterol oxidase. However, there are several oxidases that have a lack of isomerization activity. They don't flip that double bond whenever they oxidize that hydroxyl group. And that results in a molecule, a metabolite, that isn't lytic to the cells. So we wanted to test the hypothesis that reducing 7-KC in the lysosome would have an impact on cell viability. So the first thing we had to do, of course, was test this cholesterol oxidase. And we found that it was active against 7-KC, although, as you can see, it's much more active against cholesterol, several-fold more less active against 7-KC. We did HPLC assays, and we saw complete degradation. This is the 7-keto-cholesterol peak, and we saw complete degradation with the appearance of a couple new peaks. And we performed a GC-MS analysis just to make sure what was going on. This enzyme had been previously characterized against cholesterol. Since it lacked isomerization activity, it was known to result in an autoperoxidation of the molecule. So we didn't know if the same would hold true with 7-keto-cholesterol, which is why we did the GC-MS. 7-keto-cholesterol does not undergo any further autoperoxidation after oxidation by this enzyme. And so here's just the basic mechanism. there's a loss of a hydrogen, the ketone is formed here, and there's no isomerization activity that's happening. So importantly, we wanted to test the effect of this on cell viability. But we needed a way to introduce this particular enzyme into the lysosome. So we used LAMP1, which LAMP1 is a lysosomal associated membrane protein. And what I did was I took this particular protein, I fused GFP to the cytoplasmic tail here, and we deleted the intraluminal portion of this enzyme, and we just inserted our cholesterol oxidase in here. And then we overexpress these in fibroblasts. And this is just a schematic showing. So I actually saved the signal peptide on the LAMP1, and then a small linker region and transmembrane domain region. We also tried a few other constructs that we generated. We overexpressed sterol-27 hydroxylase. We overexpressed the same cholesterol oxidase in the cytoplasm instead of the lysosome. We overexpressed 11-beta hydroxysterol dehydrogenase. We also used that LAMP1 by itself. We thought to use that originally as a control. And we overexpressed M-cherry as well, which is just fluorescent protein as a control. And these are some bright field microscopy images that we took of the results. And so what you're seeing here in this first column is untreated but transfected cells. So these cells were transfected with the LAMP1 cholesterol oxidase fusion protein construct. These were just transfected with LAMP1, and these were transfected with that sterol-27 hydroxylase. And remember, the metabolite of this particular enzyme is almost completely non-toxic to the cell. However, what we saw when we looked at concentration of 50 micromolar of 7-KC transfected cells or treated cells, we see that there's still a good cell structure here, very similar to the unamended population of cells. This is just some fluorescence microscopy showing the fluorescence, so we knew that GFP was being expressed in the cell. However, this was kind of a surprise to us. The LAMP1 construct, we see very dense. We didn't see a lot of toxicity from the transfection, which we normally do. But at 50 micromolar, in our images, these look like very healthy cells, especially even in comparison to our construct. However, on the other hand, the CYP27 or the sterol-27 hydroxylase, this is what we would normally see just in non-transfected cells. We see almost complete cell death at 50 micromolar 7-KC. So, but we definitely did see a lot of surviving cells at this concentration with our construct. And this is just an image quantifying our data. This would be the baseline because all the cells are basically dead at this point. But what we noticed is that, in contrast to what we saw on, under the microscope, we actually saw very, very high viability in our LAMP1 cholesterol oxidase construct, actually more so than any other treatments. So of course we repeated the experiments, a number of times, and we saw the same sort of story each time. And even in the bright field microscope images look the same too. So we're still trying to work that out, but we do see a protective effect with our construct. A couple other of our treatments were protective, but the LAMP1 was significantly protective at a concentration of 37.5, but oddly, not really any of the other concentrations. Our cytoplasmically expressed cholesterol oxidase was protective, but not very much so at 37.5 and 50. So what we saw here basically is that we did see much higher cell viability in these cells that were transfected with our construct relative to the other treatments. Now this is these are confocal images of the cells that were transfected with the construct in comparison to cells that were not transfected. And what you can see here, it's a little blurry, but we do have, number one, we do have very, very good localization of our construct with the GFP. Remember, there was a GFP tagged to this particular protein in the outer membrane. So we know it's localizing properly in the lysosomes. And we still do see lysosomes clear, so we don't really think there's much lysosomal membrane permeabilization going on, but we do have morphological differences from untreated cells. And there's a couple explanations we've thought of for that. One could be that we're having unintentional transformation of cholesterol. And even though cholesterol, at least we think this metabolite isn't lytic, there could still be some different membrane properties that are resulting in some morphological differences. The other answer could be that since we actually mutated LAMP1, which is a membrane protein, and we overexpress this, we could be saturating the lysosomes with this modified LAMP1 that is for some reason causing these differences. However, we haven't figured that out yet. This is work that we're ongoing trying to determine. We also did try cyclodextrin treatment. So, of course, increasing 7-kc efflux was an important part of our strategy. What you can see here is these are the M-cherry controls, and this is cytoplasmically expressed cholesterol oxidase. And here we're seeing almost complete cell death, so we really didn't see any benefit from this, and M-cherry as well. However, with our 0.9% cyclodextrin, we saw almost complete cell protection at 50 micromolar 7-kc. And here is just, this is quantified. You see all the way out even to 100 micromolar 7-kc, we're seeing a very protective effect using 0.9% cyclodextrin. If you go too high in the concentration, you see cytotoxicity all the way from 0, just from the cyclodextrin itself. So that was that. So some of the work we're looking at now is we're combining treatment, cyclodextrin, with perhaps some of the cytoplasmic or mitochondrial enzymes to increase 7-kc efflux out of the lysosome, where we can actually transform it in the cytoplasm, where we could have redox enzymes that could hydroxylate the substrate. We're also looking at engineering substrate specificity in this oxidase to see if we can reduce activity against cholesterol and increase activity against 7-ketocholesterol. And we're also going to be working some more with LAMP1, since we found that LAMP1 does have a protective effect. It actually has a role potentially in cholesterol transport and also in stabilizing lysosomal membranes. And some other projects we're working on are, one of them we're working on is we just started a project in lipofuscin degradation. Rob O'Callahan in our lab is working on that, and he's going to be working on some mammalian and yeast-based models of lipofuscin accumulation in the lysosome, specifically using a master regulator of lysosomal biogenesis. So with that, I'd just like to acknowledge my lab, boss Pedro Alvarez, Laura Segatori at Rice University, of course the SENS Foundation for providing funding for this research, as well as collaborators at the University of British Columbia, Bill Nunn and Lindsay Eltis.
Thank you very much. Maybe we have time for one question.
Hey.
Hi.
So, of course, cyclodextrin is used as a pharmaceutical carrier for all kinds of reasons. Is it, I mean, the pharmacology, the pharmacokinetics must be really well known. Are those kinds of concentrations of cyclodextrin in target cells across the body achievable with oral dosing or even intravenous? Or
Yeah, so the toxicology it's very non-toxic. It's used in everything from Febreze to pharmaceuticals and stuff. And this is just our first kind of pass with cyclodextrin. I don't know if lower concentrations. We only tried 2 concentrations, and this was just based on the maximum solubility of cyclodextrin in water. So we're going to play around with the concentrations a lot more. There are some people that have been using cyclodextrin in humans. So there's a woman, I don't know if you're familiar with her, whose children actually had Niemann-Pick disease. And she is using cyclodextrin. She started using dietary cyclodextrin on her children to treat Niemann-Pick, since there's no cure for that disease. And she was successful with it. However, cyclodextrin can't cross the blood-brain barrier. So she actually was able to get a clinical trial to intrathecally inject her children with cyclodextrin. She's currently treating them now using that method. So there's probably a lot of data there on levels and stuff.
Well, and for atherosclerosis, getting vascular coverage is most of what you want to do anyway. So
Yeah, correct.
Well, thank you. Okay, we'll take one more question.
It's more of a comment, but if you overexpress LAMP1, it's going to result in lysosomal exocytosis, because excess levels of LAMP1 results in the LAMP1 associating with the plasma membrane, and the lysosomes then dock with the plasma membrane and fuse with the cell membrane, and the contents are released extracellularly. So I think what could be happening is that your cells containing 7-KC, the lysosomes are getting exocytosed, and so it becomes protective because they're eliminating the 7-KC extracellularly.
Yeah, that could also be the effect that cyclodextrin has, as well as increasing exocytosis. There have been a couple papers. One says it increases exocytosis. The other says it facilitates diffusion across the lysosomal membrane. LAMP1, I think, is somewhat the same. There have been a couple papers published showing that LAMP2 is involved in cholesterol transport in the cell. So I was kind of surprised that LAMP1 did have an impact. But I've actually just recently contacted the authors of the papers that were talking about LAMP1 and cholesterol transport. And they have actually found a role of LAMP1 in cholesterol transport as well. I don't know if it's a combination of the two, if one of those roles is predominant, but hopefully we'll figure it out.
Okay, thank you very much.
Thank you.